Let's talk about DNA distribution within the nucleus today. It's really important to understand how DNA is localized because its transcription depends on where it is in the nucleus. So the nucleus is a sphere, and we call this sphere the nuclear envelope. And normally the parts of the chromatin, which is DNA, that are closer to the nuclear periphery, so closer to the nuclear envelope, closer to the edge of the nucleus, those parts of the chromatin is usually heterochromatin. It's that tightly packed DNA that doesn't really get transcribed. So everything that's on the periphery, that's normally silenced. It is not active, it doesn't get transcribed. Those are normally the telomeric regions of the DNA and the centromeres, those median parts of the chromosomes which contain a lot of repeats that we talked about earlier. So those parts are on the periphery, so one's in the middle. In the middle we have euchromatin. Those parts of the chromosomes that need to be transcribed, normally those parts would contain the genes that need to be active at this point. There are some heterochromatin regions in the middle of the nucleus, and those heterochromatin parts are, well, that's those genes that need to be silenced in this particular cell type. Basically, this type of heterochromatin, and it's called facultative chromatin, heterochromatin, that's that heterochromatin that is responsible for silencing, say, liver genes in a neuron. You get the idea, right? So we don't want those genes to be active because we want the cell to be, a neural cell, right? The type that we want, and we need only those genes that are appropriate for this type of cell to be active in this type of cell. So everything that belongs to other cell types, that's silenced using heterochromatin. So it's tightly packaged, and the transcription machinery cannot really get close to that, get inside this region and start transcribing those genes. So that's the major idea that I want to explain here, is that everything that needs to be silenced, that's on the periphery, and the active genetic regions that need to be transcribed are in the middle.